Hello everyone. In this video we're going to explore how muscles contract from molecular level from inside of a sarcomere to different types of muscles and reflexes and how they all work together to create movement. Um, we'll also look at where energy comes from to power these kind of contractions. Okay, let's begin with skeletal muscle type. This is ones that we control voluntarily. Let's see. Let me just do a quick drawing. I'll make it fun. Okay. This is a skeletal muscle. This is a muscle fiber, and this is a sarcomere. That's the sarcomere. It's the functional unit of the muscle fiber. It's defined between two regions, but it's defined as the region between two Z-lines, Z-line, Z-line. Within it, there are two types of filaments. Thick filaments, that's made mostly of myosin, and thin filaments, that's made mostly of actin. The thick filaments will be in the center, and the thin filaments will extend from the lines. Myosin is what thick filaments are made of, and it's kind of looks like a golf club. Check it. It kind of looks like a golf club with two heads that can pivot. Actin is like it will form a twisted. Okay, I can't do it like that. Jeez. Actin will form like a twisted double strand. And then wound around actin are two regulatory proteins, tropomyosin, which covers the binding sites, and troponin, which moves tropomyosin out of the way when calcium binds. Okay, that's skeletal muscle. Now we're gonna move. Now what we're gonna do is we're gonna go from a nerve signal to muscle contraction. It all happens with an action potential. So I'll go like this. I'll use my muscle fiber. There's a motor neuron, and it connects to the muscle fiber. It's called the NMJ, or the neuromuscular junction. It releases acetylcholine, which binds to the sarcolemma, and that's in the muscle cell membrane, and that causes depolarization. The action potential goes throughout the T tubules, and triggers voltage-sensitive proteins called DHP receptors. These are physically linked to rhinanodine receptors and sarcoplasmic reticulum. It's in muscle cells. When these are activated, calcium floods out of the sarcoplasmic reticulum and into the cytoplasm, and that's a signal for contraction. Right here, right here. Once calcium is released, it binds to troponin, the thing from before. That's on actin. And then that moves tropomyosin away, that's covering the binding spot, and that exposes these binding spots of actin. I'll draw a little. We've got troponin and tropomyosin. Troponin, tropomyosin, calcium comes and binds to troponin, which moves tropomyosin away, exposing the binding site. This is when the myosin head will come and attach to troponin, to actin, and it forms a cross bridge, which uses energy from ATP, and the myosin head pivots and pulls the actin toward the M line, and that shortens the sarcomere, and that's what contraction is. Okay. Muscles need to keep contracting, and there's multiple ways to do that. We have an energy source. I'm going to draw a little table. Energy source. And the speed. Duration. And then, yeah, that's good. Okay. A stored ATP. Stored ATP. It's very fast and can last about 2 seconds. So, not very long. This is just kind of like the initial contraction. It's not supposed to last long. And it's also, there's not as much supply. We don't like to use a lot of ATP. Creatine, phosphate. This one is pretty fast. And it's about 10 seconds. This one is like sprinting, and it's good because it can replenish ATP very quickly, but still doesn't last very long. Then we have anaerobic glycolysis. This is a moderate speed, and about 30 to 60 seconds. This is like intense activity. Yeah. Intense exercise or something like that. And then finally, we have aerobic respiration. This is very slow, and it's long term. You can go for a long time. This is like jogging or standing or something like that. Most efficient. There's the chart. Memorize it. So short bursts will rely on stored ATP, increasing phosphate, where longer, more moderate activity will do things like anaerobic glycolysis or for endurance, um, aerobic respiration. Okay. Now we are at smooth muscle. Now, not all smooth muscle is skeletal, so it's not all muscle is skeletal. So let's look at smooth muscles that are found in organs and blood vessels. Um, in a single unit smooth muscle, one cell's action potential spreads to others through gap junctions, like in intestines. And multi-unit is which each fiber is individually controlled, like in the iris in your eye. When calcium enters, it binds to calmodulin, which activates MLCK. MLCK phosphorylates myosin, allowing it to bind to actin and contract. Relaxation happens when calcium is pumped out and the myosin light chain is dephosphorylated. Smooth muscle is much slower, but it's more energy efficient. This is like for maintaining organs. It's for smooth muscle. It's involuntary. This is involuntary actions. The calcium comes from extracellular and sarcoplasmic reticulum. Instead of troponin, we have calmodulin. So yeah, and also it's much slower, like I said. Okay, now finally, let's look at how muscles sense and respond to stretch. Um, Muscles with muscle spindles will detect stretch and trigger the stretch reflex. That's a muscle spindle. And this is the tendon. Muscle spindles will detect the stretch and trigger the stretch reflex. And this causes the muscle to contract without resisting overstretching. The Golgi tendon organs will sense the tension and trigger relaxation to prevent damage. So it can happen a lot. You can have you can stretch your muscles a lot more than the Golgi will stop you. The Golgi usually stops you. So we'll have the reflex arc would be it starts with sensory, and then it goes to spinal cord, and then it goes to motor neuron, and then it goes to the muscle. In the stretch reflex, the signal never really reaches the brain; it just goes to the spinal cord first. It's much faster. The withdrawal reflex is similar. It pulls the limb away from something painful, like touching a hot stove. That's what the Golgi tendon is for. So to summarize, the skeletal muscle contraction starts with the calcium release and ATP-driven cross-bridge cycling, and is powered by several energy systems, as we looked in our table. Smooth muscle contracts differently through the I'll say it right one time, calmodulin, and MLCK. Muscle receptors will make sure that we move safely and efficiently. That's what I got.